Hi, so today I will be going over the lytic and the lysogenic cycle. So what is a bacteriophage? A bacteriophage is a virus that infects a bacterial cell. Okay, so now we have that settled. Now let's move on to the lytic cycle. Now when I say the word lytic, it kind of sounds like lysis, right? Well, that is because lysis is the end result of the lytic cycle. The bacterial cell lyses, okay? So now, what happens? Number one, attached. The bacteriophage, it lands on the surface of the bacterial cell. Number two is penetration. So the bacteriophage's RNA or DNA goes inside the bacterial host cell. And then number three is biosynthesis. DNA replication, protein synthesis, all occurs, but it occurs for the viral DNA. So think about all of the new DNA strands being made, all of the new proteins being made, but that is all for the bacteriophage, which leads us into the next stage, which is maturation. These proteins and these DNAs, it's all being made to actually grow bacteriophages inside the host cell. So the new phage particles are all being assembled, which leads us to the next stage, lysis. The cell just literally explodes. All of the newly made bacteriophages, they all escape and they all continue this cycle. Now we're going to move on to the lysogenic cycle. So the main difference between the lysogenic and the lytic cycle is that in the lysogenic cycle, we do not really go through biosynthesis. So number one, attachment. So the bacteriophage, it lands on the host cell's surface and then it attaches itself. So again, the DNA or RNA of the bacteriophage will enter the host cell. Now, this is the main difference. So instead of biosynthesis going straight into DNA replication or protein synthesis, lysogeny occurs. Lysogeny is when the viral DNA or RNA is being integrated into the host cell's DNA. Within the bacterial DNA, there's also an integrated segment of the viral DNA or RNA. And guess what? Since the bacterial cell is a living thing, it is going to reproduce through cell division or binary fission. And in binary fission, it reproduces really quickly at mass amounts. And guess what? The same DNA is being used. So all of the newly made cells are going to have that viral DNA in it. What happens next is that it remains dormant until the right condition has been met. When there's, for example, nutrient depletion for the bacterial cell. So what happens is that the integrated viral DNA or RNA gets excised from the bacterial DNA. So it goes from this to that. And what happens is that it enters the lytic cycle. So that is it for the lytic cycle and the lysogenic cycle. If you have any more questions, please comment down below what topics I should do next or, just anything in general. But thank you so much for listening and goodbye.